Water is split by photosystem 2 on the side of the membrane facing the thylakoid space. As a plastocyanine, PQ, transfers electrons to the cytochrome complex, 4 protons are translocated across the membrane onto the thylakoid space, and a hydrogen ion is removed from the stroma when it is taken up by NADPA+. Notice that in step 2, hydrogen ions are being pumped from the stroma into the thylakoid space. As in figure 10.17, which is the thylakoid membrane. The diffusion of H plus from the thylakoid space back to the stroma along the H plus concentration gradient powers the ATP synthase. These light-driven reactions store chemical energy in NADPH and in ATP, which shuttle the energy to the carbohydrate-producing Calvin cycle. The second stage of photosynthesis, which takes place in the stroma of the chloroplast, can occur without the presence of sunlight. In this stage, known as the Calvin cycle, carbon molecules from CO2 are fixed into glucose. The reactions of the Calvin cycle is as follows. A 5-carbon sugar molecule called ribulose biphosphate, or RU-BP, is the acceptor that binds CO2 dissolved in the stroma. This process, called CO2 fixation, is catalyzed by the enzyme RUBP carbolaxlase, forming an unstable 6-carbon molecule. This molecule quickly breaks down to give 2 molecules of the 3-carbon 3-phosphylglycerate, also called 5-glyceric acid, PGA. The second step is 2 3-PG molecules are converted into glyceraldehyde 3-phosphate molecules, a 3-carbon sugar phosphate, by adding high-energy phosphate group from ATP, then breaking the phosphate bond and adding hydrogen from NADPH plus to H plus. 3 turns of the cycle using 3 molecules of CO2 produces 6 molecules of G3P. However, only one of the 6 molecules exits the cycle as an output, while the remaining 5 enter a complex process that regenerates more RUBP to continue the cycle. 2 molecules of G3P, produced by a total of 6 turns of the cycle, combine to form 1 molecule of glucose.